Hello, everyone. Welcome to GenScript 2024 webinar series. I'm Claire Zhou, the product manager for our CRISPR services at GenScript. It's my pleasure today to introduce Dr. Sandra Miller as our guest speaker. Sandra is an assistant member in the Department of Cell and Molecular Biology and founding director of the Center for Advanced Genome Engineering, as well as the associate director of Shared Resources for the Comprehensive Cancer Center at St. Jude's Children's Research Hospital. Sandra completed her PhD in cell and molecular biology from the University of Texas Southwestern Medical Center in August 2008. While at UT Southwestern, she worked in Matthew Porter's lab on the optimization of zinc finger nucleus for use in mammalian cells. After graduate school, she was recruited to Sigma as a senior scientist in R&D, working on the composer zinc finger technology. In 2012, she returned to academia as a founder and director of the Genome Engineering and Activity Assay Center at Washington University School of Medicine in St. Louis. In 2017, she joined the faculty at St. Jude's Children's Research Hospital, where she established as directing the Center for Advanced Genome Engineering. Sandra has overseen the creation of 1,000 custom edited clonal cell lines, more than 250 custom edited preclinical animal models, and over 50 custom pooled gRNA libraries. In today's webinar, titled Functionalizing Genome Editing for a Broad Range of Targets, Dr. Miller will discuss what is the best practice for creating knock-in cell lines, how large insertions can be achieved with good efficiency via direct embryo engineer injection and single-stranded DNA, as well as how NGS analysis enables the detection and quantification of CRISPR editing outcomes on a diverse range of sample inputs, including cell pools, clonal populations, and animal tissues. If you have any questions during the presentation, you can submit them by typing them in the question box that you see on your screen. We'll have a Q&A session at the end of the talk. So with this, I will pass the presentation over to Dr. Miller.
Great, thank you so much for that kind introduction, and I'm happy and thankful to have been invited to give this presentation. It's always great to talk about what we're doing, and although I'm in my office, I feel like we're at least somewhat together if we are communicating science. So yeah, my name is Chandra Pruitt-Miller. I publish under Pruitt-Miller, so if you're looking for any of my papers, it'll be under Pruitt-Miller. And I am the founding director of the Center for Advanced Genome Engineering at St. Jude Children's Research Hospital, and we call ourselves the CAGE. And our missions in the CAGE are basically threefold. First, we help others do genome editing. So we facilitate investigator-initiated projects, so we help them do genome editing. And then second, we spend about 25% of our time doing independent R&D projects so that we can be at the forefront of this fast-paced field. And then finally, we support key initiatives such as the gene and cell therapy programs at St. Jude Children's Research Hospital, and specifically on CAR-T and sickle cell disease. So as I mentioned, we spend 75% of our time doing helping other people do genome editing, and then 25% of our time doing independent R&D. In that 75% of our time, we basically provide 6 main services to St. Jude investigators, and I'm going to list them here so you can kind of get an idea of kind of the scope of what we're doing in terms of genome editing. So in those 6 services, we have design, assemble, and validate guide RNAs. We do this both in RNA format, so RNPs, and then also in plasmid format. So if people need to use something like lentivirus or primary cells, we can also assemble those and validate those. We also create custom clonal cell lines. This can be pretty much any modification in any nuclease amenable cell line. We also create animal models, help with generating animal models. These are typically rodent models, mouse or rat. And we also do targeted next generation sequencing for CRISPR edited samples. We also facilitate high throughput genetic screens by creating and QCing pooled guide RNA libraries, and then also helping on the back end to analyze those screens. And then we also create edited cell pools. And this is basically for those folks who don't really need a single cell derived clonal population, in which we can create a highly edited cell pool. So that's kind of the scope of what we're doing on our core side of the business, or core side of the operation. But we also, I wanted to give you kind of an idea of numbers, how much of this we're doing. So with guide RNAs, we stopped counted because all of our projects start with a guide RNA. In terms of cell lines, just in the last 3.5 or so years, we've created 522 custom cell lines. 93 animal models against mostly mouse. And we do about 4,000 samples per week with targeted next generation sequencing. So we do a ton of next generation sequencing. And so I'm going to walk you through how we use that in our workflow. And then we've also created 36 custom pooled libraries, and we've analyzed even more downstream. And then just since January, we started offering these edited cell pools to St. Jude investigators, and we've created 59 edited cell pools. So I want to put this slide up just to make sure we're all on the same page, and then also to mention that we are using traditional genome editing kind of in our core operations. And when I say traditional genome editing, I mean that we're using a nuclease-induced break, typically with SP-Cas9, to make a double-stranded break near the site of modification. And then we rely on the cell's endogenous natural repair pathways to repair that double-stranded break so that we get the targeted mutagenesis that we want. The two major repair pathways by which double-stranded breaks are repaired in mammalian cells are non-homologous end joining and homology-directed repair, or NHEJ, shown in green, and HDR, shown in red. So depending on how the cell repairs the double-stranded break that we recreated with our nuclease, you can get all kinds of different modifications. You can get gene disruptions, you can get direct ligation, deletions between two cuts, inversion between those two cuts, translocations between cuts on different chromosomes. And then if you supply the cell with an extra chromosomal donor template, you can get precise single nucleotide modifications or even large insertions that are precise. I also want to point out on this slide that because we're using traditional genome editing, we are relying on that double-stranded break to occur. So we need to be able to make those double-stranded breaks very efficiently. And I also drew this arrow larger than this arrow because non-homologous end joining is the preferred pathway of repair in mammalian cells. And what this looks like is you typically get these things, these gene disruptions. So these are basically a few base pairs added or deleted at the cut site. We call these insertions or deletions indels. And so that will be important for later on in the talk. I also want to mention that we are making those double-stranded breaks, and so we typically, for the operations side of our group, we use SP or S-pyogenase Cas9. And just again, so we're all on the same page, Cas9 is basically a two-component system. So we have in blue, I'm showing the Cas9 nuclease, so it's going to make a double-stranded break in genomic DNA. And then in this purple and red color, we have what we call a guide RNA, which is an RNA molecule that you can basically guide the Cas9 nuclease to a specific site within the genomic DNA. And the way you change out the guide RNA is you basically just swap out these 20 base pairs at the 5' end of the guide RNA, and they will basically, depending on what the sequence is here, you can redirect where the nuclease is going and making the targeted double-stranded break to start that repair pathway to get the targeted mutagenesis that you're looking for. All right. So because we need a double-stranded break in order to start the genome editing process, we're using traditional genome editing, we basically need to have a good assay for assaying for gRNA activity. And there's actually several different assays that folks use to see if their guide RNAs are active or giving them a double-stranded break, but they all start out in a very similar manner. In the first step, you basically take your guide RNA plus Cas9, you deliver that into cells. Some of those cells will get double-stranded breaks and some will not. Then you harvest the genomic DNA from your cells. You do a PCR around the modification site, shown here as a red X. And then you do one of these downstream applications or assays to figure out if you've created those small insertions or deletions at the cut site. So I've kind of just grouped them into 4 different groups. So there's this T7E1 or cell 1 assay, targeted next generation sequencing, TIDE or ICE, and then there's this assay called IDA. And so for our group, because we're doing so much genome editing on a broad range of cellular targets, we wanted to figure out what was the best assay for what we are doing, and then compare to a survey between the 4 assays. So a work led by Monica Sitmanat with the help of Sam Peters, Colin Florian, and Patrick Connolly. We published this about 2 years ago, so if you want to go look this paper up, you can, and you can see how these assays compared. But just to kind of summarize what we found, we needed an assay that would check off a couple boxes. So we needed an assay that would identify active guide RNAs, show us indel sizes and the sequences of those sizes, of those indels, and that accurately portrayed editing rates both in pools of cells and then also in clones. And then finally, because we're doing so much genome editing, we needed to make sure that we had a high throughput assay. So from these 4 assays, the only assay that really checked all of our boxes was targeted next generation sequencing. So as with any assay, all 4 of these different assays have different limitations, but because they're all PCR based, they have some common limitations, and I just wanted to walk you through those. So because they are PCR based, they have PCR bias. meaning that the smaller the amplicon is the more preferentially it will be amplified. So smaller amplicons will be amplified more efficiently than larger amplicons. Also, because it's PCR based, we can't detect all chromosomal aberrations or gross chromosomal aberrations. And this is because if you lose a primer site, you will actually lose, you won't be able to detect it by any PCR based, or you'll be able to amplify it via PCR. And then there's also an amplicon size limitation. Each of these are slightly different, but really all of them are less than 1,000 base pairs, partially because of how difficult it is to amplify off of genomic DNA and get a clean PCR product. So although there are common limitations to all of these assays, there's really one unique limitation to next generation sequencing. And it's this high entry barrier to entry. You have to have a kind of expensive machine. The reagents that you need to use to run a flow cell are kind of expensive. You need expertise. You get all of this data out, and you don't really know what to do with it. It's in FASTQ files. You get lots of files. And so we basically set out to make this efficient, and we actually use NGS for almost all of our analysis to make sure that we are getting the editing outcomes that we want. And so I thought it might be helpful to walk you through our genome editing workflow and how we use targeted next generation to make sure that we're efficiently creating the editing outcomes that we want, whether it's in cell lines or in animals. So the first thing that we do is we introduce guide RNAs. And so for cell lines, we basically transfect, we nucleofect, we oftentimes use nucleofection, but if you can deliver the guide RNA reagents or the Cas9 CRISPR reagents to the cells, that's basically what we're trying to do. So introduce them into the cells, or if you're creating an animal, you could do direct embryo injections or even electroporation. So get the stuff into the cells that you want to get in, so guide RNA, Cas9, and donor if needed. And then you basically culture the cells. So you can culture these as single cells, so we can flow sort them or FACS sort them 1 cell per well into a 96 well plate, or we can use them, assay them in a pool, so culture them in a pool of cells. And then for embryos, if you're trying to create an animal model, you basically implant these embryos into a pseudo-pregnant female, and then the next step is to harvest genomic DNA and analyze to see if you got what you wanted to get, the modification that you wanted. So this is broken up into several steps. First, you're going to harvest the genomic DNA and PCR amplify the region of interest. And we do this by a 2-step PCR method, which I'll explain more in the next slide. But basically, we first amplify the region of interest with partial Illumina adapters hanging off. And then we do a secondary PCR to put indexes on so that we can basically mix all our samples together and then demultiplex them once we have run the NGS. So you do the PCR, pull those samples together, sequence with next generation sequencing. We use the Illumina platform, and then we demultiplex, and then we analyze our data. And we use a program called CRISP.py, which I'll go into more detail in the coming slides. So to go into just a little more detail about this 2-step PCR sequencing method, I thought I would show you. Here's basically our region of interest shown in teal. And then we design primers that are specific to that gene of interest. And we have a limitation of about 500 base pairs because we typically do paired in 250 by 250 sequence reads. And so the shown in blue are the locus-specific primers. And then we have these partial Illumina adapters shown in orange on both sides. And then we follow that up with a second PCR where we basically have unique forward and reverse primers that have indexes. On the reverse, we have 10 base pair indexes. On the forward, we have 6 base pair index. And we can mix and match those different indexes to give us basically a lot of samples that we can index differently so that we can have unique indexing. And then you get a amplicon that looks like this, or a template that looks like this, that you can run on a flow cell and then demultiplex again based on those indexes and then also that locus-specific region. So after you run NGS, this is what you get. You get a whole bunch of FASTQ files. And I think this is overwhelming to a lot of people when they get, I don't know, depending on how many samples you run, you could get, 4,400 different FASTQ files. And what do you do with all that information? Inside of those files, you basically have the editing, efficiency of the editing. You have the size of the indels and you have the sequence identity. You just have to get that information out. And so for us, as a lab of biologists and molecular biologists, we really had no coding experience. But we knew what we needed in order to be able to do what we wanted with our genome editing workflow. So Patrick Connolly in the lab really took it upon himself to teach himself Python. He took a course on Codecademy online. It's a free online course, so he could understand how to write Python scripts. And he basically wrote this script. And I'm going to tell you a lot about it. It is published. We just published it about a year ago, I guess a little over a year ago now. I'm super proud of this, and I'm really impressed with Patrick that he took this initiative to do this, and it's really been game-changing for our lab. And we've actually been using this for about 8 years now, but we had so much to do, we didn't have time to publish it with all the genome editing projects that we had. So how does CRIS.py compare to other analysis programs? So there's definitely other NGS analysis programs out there. There's several. This list is probably out of date already. But we needed a couple things because of what we're doing in the core. We needed a couple things. We needed to be able to check off certain things again. So we needed to be able to run locally on our desktop computers. We didn't want to have to rely on somebody's server. If it went down, we would be stuck. We needed to be able to analyze over 20 samples and really hundreds of samples for a given loci. And then we also need to be able to search for more than one donor sequence. So sometimes we want heterozygous modifications, and we need to be able to look for both modifications that we're looking for. And then this is really the key one that sets CRIS.py apart from the other NGS programs that I'm aware of, is that we get a master summary file that is searchable and sortable. So this really helps us go through the different projects and figure out what's the best clone that we have, or what kind of different clones we have. I should mention that a lot of these other programs are superior, and ours definitely the coding, and then also their analytics as far as like what they output. They give really great graphical information, pie charts, graphs. So I'm not trying to knock any of these. It's just for what we're doing, and the functionality of CRIS.py, I think is really as simple, and it really is, if you're not a computational biologist, you can definitely understand how CRIS.py works, and it's not a black box. You can really tailor it to your needs. So I thought I'd go over how CRIS.py works. So basically you start out with your reference sequence. So remember, we're doing that two-step PCR, so we know what our amplicon is, we know where our modification is. So you use, as your reference sequence, you're going to just use your amplicon sequence. So from F to the forward primer to the reverse primer. Then we give the program what we call a seq start. So this is about a 10 to 14 base pair sequence at the 5' of your reference sequence, or that amplicon. And then we also give it what we call a sequence end, or seq end, which is a 10 to 14 base pair sequence at the 3' end of your reference amplicon. And then we say any of those, look through all of those FASTQ files, and anything that has this seq start and this seq end, we want you to measure the distance from the start to the end, and then compare it to the reference that we gave it. So we gave it the reference. Let's say in this case, our reference was 250 base pairs, and then one of our sequencing reads was 249. It would basically compare those two, and then it would report back the indels. So an indel that had no insertions or deletions at the cut site, which we would expect from non-homologous joining, would be reported back as a 0 base pair indel. If you're missing a single base pair, it would be reported back as a -1 base pair, and so on. So that's the way we use CRIS.py to look for knockouts or insertions or deletions. But we're oftentimes wanting to do knock-ins. And so what we also have is a feature where we can provide the CRIS.py program with basically an unlimited number of test sequences. So if we're looking for specific editing outcomes, we just tell the outcome, and it checks for the presence of that test sequence within the sequences that have the start seq and the start or the seq end. So CRIS.py does two things. Basically, it looks for indels based on length of the amplicons. It says, does this differ from the reference amplicon that was entered? And then it looks for exact matches to sequences that you give it. And so in terms of input, this is kind of an example project. So in this case, we have an amplicon here on the top. Our reference sequence would be from the forward primer to the reverse primer. We have our seq start here, our seq end here. And in this experiment, what we were wanting to do is to know, is guide 10 or guide 14, which bind very close to each other, which one of those is more active, or are they both active? And so what we do is, this is the command line. This is what the coding program would look like. And you basically don't touch anything except for the stuff in red. The first thing you have to input are 6 things. First is what you want to call the folder that all your data is going into. That's super easy. You can call it whatever you want. Then you're going to need to tell it the reference sequence. Again, super easy, just from forward to reverse primer. That's your reference sequence, your amplicon sequence. Then you need to give it a 10 to 14 base pair. It can be really any length, but 10 to 14 is a nice small number. Seq start and seq end. So seq start is at the 5', seq end is at the 3'. Then you're actually going to leave this most of the time alone. That's going to say that look in any files for these things that end with .fastq. And then you have this test list, and you can give it an unlimited number of test sequences that you want to look for. So in this case, we're just looking for the 23 base pairs of guide 10 and the 23 base pairs of guide 14, and we're looking for exact matches to those. So that's the input. And I'm going to pause here because whenever we submitted this for publication, a lot of the people were coders, and they said, hey, you know, it's not common to actually modify the command line. And so most people have a GUI, or graphical user interface. And so that hasn't been published yet, but what we do have, Jake Steele in the lab has actually gone through and made a GUI for this. So if you prefer to use this GUI, you can. There are some benefits to using the command line, but certainly if you want to use the GUI, it's available here. And both the CRIS.py original program and what we call PrettyCRIS.py are available on GitHub for free. All right, so let's go to the output. So I told you how you get what you have to input. You basically have your FASTQ files, then you have to tell it the reference sequence start, end sequence, and then your test sequences. And this is what you get out. This is that master summary file, and it's in a CSV format, which is basically an Excel file, which is, again, searchable and sortable, which is key if you're looking through a lot of different clones. So I'm going to walk you through this output. I'm going to kind of go step by step. So the first column is called name. That's just the well in which your sample occurs. So again, this is determined based on the demultiplexing those indexes, those forward and reverse primer indexes. And then we basically have, this is usually left empty, so you can fill this out on your own. So we inputted this name. We said negative control, guide 10 transfected, guide 14 transfected. Then in the next column, you're going to have the total number of reads. So we had 980 reads for the negative control and so on. Then the next columns are going to be whatever you put in as test sequences. So we put guide 10 for the first name of that column, and then we put guide 14 for the second column. And then it's going to tell you the number of times, the number of reads, and the percentage of reads that gave those exact matches. The next column is going to tell you the percent of reads that are not wild type linked. So basically anything above 0 is going to be considered an indel. And then the last part you're going to see is we're going to report the top, and I'm only showing 4 here, but we typically report the top 8 indels. So this is the most common indel in these samples, which was 0 or wild type link, followed by, in this sample with G10, a +1, a -15, and a -2. And then the last thing on this output is going to be some QC checks, which I'm going to glaze over for now, or gloss over for now. If you want to talk about these in the Q&A, I'm happy to talk about those as well. But easily I can go through here, I can look at this file, and I can say, okay, guide 10, we got 80.7% cutting, with meaning alleles that were not the wild type links. So I automatically easily know guide 10 was the most active, and I can tell you what fraction of indels, basically a +1 was the most common indel, a -15 was the next most common. It's very easy to look at and understand what's going on. The second output file is a text file. So this was just a summary file. It kind of told you kind of what in general was going on, but it doesn't tell you what this +1 sequence is. The second file is an output file that's a TXT file, which will give you the top exact matches to the sequencing reads in any given well. So again, this is our negative control, which was in well 1B07. And what you can see is it'll tell you this line, this sequence, is the exact match and occurred 644 times, followed by this sequence, which occurred 11 times, this sequence which occurred 10, and it could go on and on and on. And so what you can notice immediately is all of these are the same length. And so what this is these are sequencing errors. So any given base pair has a small percentage, about a 0.02 to 0.04% chance of having a miscall. And so that's what we're seeing whenever we get so many reads. And we're actually binning them based on exact matches. And so in this case, you can see these are our guide RNAs over here. So they're cutting where those arrows are, and these mutations have nothing to do with these guide RNAs. So these are what we call sequencing error. And again, I can just easily look at this and say, okay, they're all the same lengths. These are sequencing error, and the fact that I have a major read and then followed by a lot less reads. However, whenever we look at our guide 10 and guide 14 samples, you can see what I call the river. You can see that these are kind of wiggly. You can see that they're not all the same length. And you can see that they have significant numbers of reads at each one of the different exact matches. So I can easily tell you that these are the samples that had guide RNAs in them. This is my control, and everything looks good. One thing I did want to mention is that whenever we're looking at the total indels and frequencies, this is looking, again, just at length, not at sequence. And so if you have something like a 45 at 23%, what we actually found out whenever we looked at the text file is that we actually had 2 different -45 indels. And so you can actually look at what the sequences are here in the text file. All right, so CRIS.py also allows you to look at HDR events, and you can analyze for multiple different donor outcomes or editing outcomes. So oftentimes people will come to us and they'll say, hey, we want to make a modification that represents a patient sample, and they're heterozygous for this modification. Can you make a heterozygous or clean heterozygous modification? And when they say clean, they mean they don't want indels on the other allele, and they don't want it to be homozygous. And so what we did in this experiment is basically we used two different ssODNs, or these are basically just short oligos that you can have synthesized. And we call one of them a block mod, and we call one of them a block. So the block mod has the modification that they want, the C to T modification. And then we also put in what we call a blocking modification in the PAM sequence. So here's that NGG, and we basically changed that C, which is a G on the bottom strand, to a T. And we did this to prevent the gRNA from coming back in and cutting after this modification occurred, because it's not enough to have just one base pair of mismatch between the target site and the modification site. So after this modification had been incorporated, there's a chance that it could come back and cut and then knock out our modification. And then on the other ssODN, so you can see here the block ssODN, we put in just that blocking modification. So the hope here is that we can make one allele that has this block mod and one that has just this blocking modification to give us what we call a clean het, where we're not knocking out the gene. So here's our outcome, or our master summary file. So you can see this is our negative control. And in this sample, we put in our guide 6 plus Cas9 plus 2 ssODNs, the block mod and the block. And in our test sequences, we looked for the exact match to guide 6, and we looked for exact matches to the block mod and the block only oligos. And you can see we're getting editing of about 13.4% and 10.4%. Quickly, I can also say, okay, my cutting is going really well. I'm getting 52.8% of the amplicons are not the wild type length. And then I can easily tell you what the amplicons are based on frequency. Then we also can look at our text file and we can say, okay, here's our negative control. All of the alleles or all the amplicons are similar in size, and you can have one major read and a whole bunch of small numbers of reads, which means this is sequencing error. And then in our experimental or guide 6 plus ssODN treated sample, you can see that we're having major reads. These are our indels, and these are the modifications that we're actually looking for, our block mod and our block. So everything looks good. And so this is really a powerful tool. NGS as a tool is really powerful because it helps us understand, is there the needle in the haystack, so that we can go forward and look for the clones? Is it worth going forward with a laborious process of going through a single cell derived clone? If we got a result like this, we would go through, we would single cell sort our clones, one cell per well into a 96 well plate. And then we would analyze, we would do the barcoding where we would put, a different barcode combination in each well. And then we would pull those samples together, run them on a flow cell, and then we would get a master summary file for those. So what this allows you to see is that you can easily analyze single cell derived clones using this master summary file. So here we're looking at a whole bunch of different types of clones that we got out of this one experiment. So you can see we have a wild type allele on one allele, and then we have a block mod on the other. I can tell this by looking at the wild type G6 sequence. This is about 50-50 ratio. We also got some clones that were block mod on one allele and block, which is what we thought we would get, where it's 50-50 approximately. And then we also got some that were block mod. So they both had the blocking mod if they're homozygous. There are 100% of those. And we also got wild type allele or wild type clones that we could use as controls. We got wild type knockout clones, so het clones that had the wild type allele and an indel. We got knockout clones. So you basically get a whole bunch of things, and you can easily and quickly tell what are the clones that are interesting and important for your next steps. And then again, the text file that accompanies it, you can figure out where those reads, what those reads are, and what the sequence identities are. All right, so I mentioned earlier that there's these other NGS analysis programs specifically for CRISPR. And I mentioned also that this master summary file is one of the things that really makes CRISPR stand out. And I'm going to just compare this to CRISPR-So2, which is probably one of the most sophisticated and widely used NGS Cas9 CRISPR analysis programs. And it's, again, beautiful, love it, not knocking it at all. But for what we're doing, we really need this master summary file. And to show you how this really affects us, if you look at CRISPR-So2, this is an example of what you would get if you just looked at one single sample. So you get 123 files per amplicon per sample. So if you looked at a 384 well plate, which we typically look through about 384 wells in order to find a clone, you'd have 47,000 files. Whereas with CRISPR, you basically get these two files that I've described over and over, the CSV file and the text file. And basically you get 2 files per amplicon regardless of the sample number. So if you had a 384 well plate, you would get 2 files, just the CSV file and this text file. So it really makes it simple, especially if you're screening through lots of projects in a limited amount of time. So we made a tutorial video that's available. You can look at the paper to find the link to that. It's only 13 minutes, and it will be helpful if you're actually thinking about implementing CRISPR into your workflow. All right, so I told you kind of how we do our NGS, how we do our pipeline, kind of the things that we're doing in our lab. And I thought I'd just give you some tips and tricks and show you some of the things that we've learned and some of the things that I can warn you about. And so one of the things that we've learned through NGS is it's, we're working with all kinds of different cell lines. Most of the time they're, cancer-derived cell lines like HeLa cells or U2S cells, and they're not diploid. And a lot of times we have a hard time figuring out how many copies there are without doing some kind of like karyotyping or SCI analysis. But with NGS, we can actually figure this out fairly quickly by looking at the allelic ratios. So here's an example. This is one of the harder ones that we figured out. And in 2A cells, this gene actually occurs 7 times. There's 7 copies, there's 7 alleles of this. And we got this master summary file back here. I'm just showing you at the bottom. So we got basically these different indel ratios. We had a -14 at 18%, a -5 at 26, and so on. And I was trying to figure out, well, how many alleles are this? This is, it's not 4 would be 25, 25, 25. That doesn't seem to match up. And then when we looked at the actual sequence, the text file, you can see that we're getting more, like we're getting 2 distinct 4s and 2 distinct +1s. And if you do the math, really what you find out is there's 7. So 100 divided by 7 is 14. So you're getting a little bit of PCR bias because this is a bigger indel. And if you do 14 times 2, you probably had 2 -5s, 2 +1s, and 2 -4s, which we can see those distinctly. So you can determine allelic frequency most of the time using NGS reads. Also, I was surprised by how much heterogeneity there is within a cell population. So sometimes people will give us a cell line. They say, I did SCI analysis on 20, or G-band karyotyping on 20 cells, and we know that the cell line is triploid. And then we get the cell line, and then we do our clonal analysis. We look at our master summary file, and what we see is that the cell line might be mostly triploid, but there's a lot of heterogeneity within that population. So by using CRISP and NGS, we can actually give them clones back that have the most common zygosity in them. So in this example, you can see that we had triploid clones here. You can see because there's basically about 50% -4, 25% +1, 50 or 25% -2. And we're getting these allelic ratios. It's not completely 25, 25, 50, but you can see this is only done with 289 reads. These are very low reads for what we typically get. And that's why I picked this example, because you can really start to figure out the allelic ratio even with very limited numbers of reads. You can also see this is the triploid ones where we have 33, 33, 33, and so on. We also get a lot of what we call homozygous ? Clones because we basically get 1 read and it's 100%. And so we don't know if that's because it's homozygous, you've gotten the same exact modification on both alleles or all 4 alleles, 3 alleles, depending on the zygosity, or if we've lost a primer binding site because of a chromosomal aberration or a large insertion or deletion that caused the amplicon to be a bigger size than we can amplify. NGS with CRISPR, CRISPR with NGS actually allows you to also genotype pups. So we will not only genotype cells and clonal cells and pools, we also genotype the pups that we make. So we help people make these animal models, mouse and rat. And a lot of times these are made by direct embryo injection or electroporation. And you can see that you get mosaic animals. If the cut doesn't happen at the single cell stage, it can happen at the 2 or 4 cell stage, and you can get mosaicism within the animal. So you don't just have 2 alleles, you have more than that. And so in this example, I've highlighted in red the ones that are out of frame. So if I was wanting to make a knockout animal, I could quickly go through here and say, okay, I want one of these animals that has a red, and maybe I'll pick A4 because it has 2 different out of frame mutations and I can get 2 different animal models out of it. Another thing that's really neat that I think we started doing, we call this a fitness assay, and we do this to determine if a gene has a dependency on a cell line has a specific dependency on a specific gene. So in this case, we're trying to figure out, does this cell line NOM6, is it dependent on this gene, gene X, for survival? And so what we'll do is we'll take a wild type sample, we'll compare it to 2 different guide RNAs, so guide RNA 3 here, and then we'll look at day 3, day 5, and day 10 post-transfection, and we'll look to see if the in frame, sorry, I don't have it shown here, if the in frame, which is in light gray, or the out of frame indels, the ratio changes. So if it doesn't change over time, then you would say, no, there's no dependency on this, or it doesn't look like there's a strong dependency. However, if you look at gene Y and you do the same assay, so again, we're looking at 2 guides, guide 4 at day 3, day 5, day 10, and guide 8 at day 3, day 5, day 10. You can see there's a strong dependency in NOM6 for this gene. And you can see that because it goes down, the out of frame, the black, goes down really actually dramatically as the time goes by. Because the cells are dying and we're losing those out of frame mutations. So that is basically a way we can figure out if a cell line if you can make a knockout cell line, and or if you're following up with a knockout screen to see if that cell line is actually dependent on the modification or the gene. All right, so we also do things like creating large deletions. So people will come to us and say, you know, in this patient sample, we have a 140 base pair deletion. We want to make a heterozygous modification. So we want you to literally cut out 140 kb. So this seems like a big task, but what we'll do is we'll put in 2 gRNAs, one here and one here. So this is the pink region we're trying to cut out. And then we have primers that we designed, we call it genome forward and genome reverse. And these primers are going to be about 140 kb apart if the deletion has not occurred, so we won't get an amplicon. If the amplicon or the deletion has occurred, so this is showing a perfect deletion where the guide RNAs just rejoin, you're going to get about a 350 base pair amplicon, which you could A, run on a gel to see if you have it, or B, you can run it on by NGS because it's under that 500 base pair limitation. So something else that is a trick that we use is we use something called bridging ssODNs or bridging oligos. And so I mentioned that if you have a perfect deletion between these 2 guide RNAs, you basically would have something that looks like this. But what we've done is we use oligos that have short homology arms, so about 40 to 60 base pairs left and right of the modification site. And an experiment I'm going to show you is we've stuck an ECOR1 site so that we can distinguish between editing events by just rejoining and editing events that have used the ssODN. So in this experiment, we did our NGS. Here's our output, our CRISPy output, our master summary file. The samples are with ssODN and without ssODN. And what you can see is we didn't get any perfect deletions in the pool of cells. So we never got a precise rejoining of those 2 ends, which was a little bit surprising to us. We do sometimes see perfect rejoining, but in this case, we did not, probably because of microhomology at the cut sites. But we did see that 32% of the deletion products had our ECOR1 site, so they used that ssODN. I'll also mention that this was the most common type of indel, distinct indel. You had a lot of other indels, you know, to make up the other 70%. But the 6 base pair insertion, which is the ECOR1 site showing up here and here, was the most seen indel, the preferred indel. So that actually helps. And then we wanted to say, does this actually, you know, okay, so 30% of them actually have the ECOR1 site, but are they actually increasing our chances of getting the modification that we want? And in fact, they did. So we screened. We sorted 4 plates with no ssODN and 4 plates with ssODN, and then we counted the number of clones that had the deletion, and then also how many of those clones were homozygous. So you can see we got about 3-fold more clones that had the deletion, 30% versus 10, whenever we added the ssODN. And then when we looked for homozygous deletions, we actually got, you know, a great increase. Almost 5% were homozygous when we added the ssODN. So that bridging oligo is definitely a trick that you can consider doing. Pretty cheap trick too. Another thing that we do is we use single-stranded DNA. So I say ssODN if it's something that's less than 150 base pairs, and I'll say ssDNA if it's something that's bigger than that. And so with recently, folks like GenScript made this available where you can actually get ssDNAs that are larger, so that we can put larger insertions in. Before this, we were really using double-stranded plasmid DNA to do this, and that's not a very efficient way of making knock-in animals. So when you're doing those injections into embryos or electrophorations, it's really not a very efficient process to use a double-stranded donor template. The single-stranded repair pathway seems to work much more efficiently, especially in embryos. So this is a big advance for the field whenever we were able to get these longer ssDNAs. So in this example, we're using an ssDNA. We're going to insert this T2A CRE. It's about 16 base pair insertion size. We have a homology arm, a left homology arm, and a right homology arm of about 200 base pairs. And then we're going to be looking for junctions. So we're going to have a genome-specific primer and then one that lands inside the junction. So this 5' junction should only give an amplicon if we have the targeted integration that we want. Similarly on the 3' side as well. So because we can use these shorter homology arms with ssDNA, we can actually directly sequence these junctions by targeted next generation sequencing. Whereas with double-stranded plasmids, we typically have longer homology arms, which makes it A, harder to genotype, and B, we can't do direct NGS sequencing of those amplicons. So here's our outcome. One of the things that should be, I haven't told you the answer yet, but basically this is done by embryo injection, and we actually looked at 15 different pups, and of those 15, 8 actually had the targeted integration by either a 5' or 3' junction positive band on a gel. So this is over for 50% of the animals that were born actually had the modifications. This is not something you would have seen with double-stranded DNA. So this is a big advance for ssDNA. We looked at just the 3' junction. I'm going to show you the data for the 3' junction by NGS. So this is our master summary file. And one thing I do want to point out about ssDNAs is that sometimes you don't get the repair that you expect, even though it looks good on a gel. Because of the way ssDNA is repaired, naturally, you don't always get what you want. So in this case, when we looked at our 3' junction, our test sequence for the 3' junction is just going to be going across the junction between the homology arm and the insert that we put in. We saw immediately that these two pups, even though they had both a 5' and 3' band, they didn't have an exact match to the junction that we wanted. So we knew something was off. But we did see that pup 4, and there's actually several pups that have the junction that we wanted at both the 5' and 3' ends. When we look at the analysis, the text file, and we can do an alignment to our read, we can see that in pup 1, we actually see that there's a 1 base pair substitution, which really all it does is it changes this last amino acid. So that's probably still a viable animal that we could use for our project. However, if you look at pup 2, you can see that there was a 2 base pair insertion, which causes the stop codon to be moved down to here. So you're actually going to be adding several amino acids on. So it's important not only to look at the master summary file, but also to look at the sequencing. And again, if you had looked at this just by gel, you might have said, okay, these are great pups, started breeding them, and then you would have had a headache whenever you started to genotype these or try to get the phenotype. I do want to also point out something about ssDNAs. I definitely recommend sequencing all the way from homology arm to homology arm, including your insert, because it does seem like sometimes we get mutations that we can't explain. These mutations around the cut site, we think is because of the repair process of the double-stranded break being repaired by the ssDNA. Same thing with ssODNs, but we think we seem to get more, I call them synthesis errors, from longer ssDNAs than we do from shorter ssDNAs. So this is an example of these two. We don't think that that came from the repair. We think that probably was on the ssDNA in the first place. But the good news is with ssDNAs, you get quite a few animals typically, and you typically can get multiple animals that have the modification that you want. So in this case, these two animals look perfect, and here's the reads that match perfectly. But I do encourage you to sequence all the way from homology arm to homology arm. With NGS, you can actually look at, and CRISPy, you can look at pretty much any kind of modification. I call it platform agnostic. So you can look at base editing, you can look at Cas9 orthologs. We typically use SPCas9, but we've also used Cas12a. We typically use Cas12a if we can't find an NGG near the modification site that we want for Cas9. Cas12a actually has a T-rich PAM, so it allows us to go into sites that Cas9 may not be able to go into. And so here's an example of that. In this experiment or this project, we wanted to insert a HIBIT tag right before the stop codon, but we couldn't find a unique NGG PAM. And so we went with this TTV here that we found in the sequence right here is the PAM. We use a short ssODN or oligo with short homology arms, and we put that in. We included a blocking modification in the PAM, so at this side, because it's at Cas12a, and we were able to put test sequences that easily allowed us to quickly tell, okay, this is the modification that we want, and the antisense oligo worked better than the sense oligo, which is what we would expect with Cas12a. You really want to use a non-targeting strand for your ssODN. All right, so I told you earlier there's some limitations to doing NGS, and I just thought I think it's important to know what they are, see what they look like, and understand them, and then also we've come up with a few ways of overcoming them. So this first limitation was PCR bias. So I mentioned that you have bias for smaller amplicons, so larger indels will be preferentially amplified. So we thought this was a good example of PCR bias when you look at the F1 generation of animals. So in this experiment, we basically started with an embryo injection. We found a mouse in the F0 line. Basically, the F0s had this 33 base pair deletion, but they were mosaic. So they had not only the modification that we wanted, but they also had other indels going on inside the animal. So they didn't just have 2 alleles that they could pass on, they had more than 2. But when you back cross that to a wild type animal, you basically have, this animal can only give 1 allele and this animal can only give 1 allele. So we know that in the F1 generation, we should have a 50-50 ratio for the modification. And if we look at our master summary file, what we see is here's the 33 base per indel. Because it's a larger deletion, so a smaller amplicon, we're having preferential bias for that. So we're getting more of these amplicons than these that are wild type links. And that's because this is about a 10 base pair or 10% difference in size of the amplicons. So that's just something to be aware of. So if things aren't making complete sense, it may be because you have a larger indel. And I would say a larger indel is like 10 base pairs or more starts giving you bias whenever you're talking about amplicons that are less than 400, 500 base pairs. Another limitation is this amplicon size. I mentioned this is kind of for all of them, but in GS we're really limited with Illumina. We're limited to 500 base pair amplicons, but you can get creative to get around this. So if you're doing a larger insertion, In this case, we're doing it in cell lines, and we were using double-stranded DNA with these 800 base pair homology arms. We can't sequence this directly on a sequencer because 900 base pairs is over that limitation. So we would want to know the 5' and the 3' junction, as I described earlier. What we do is a second PCR where we do this first PCR and then we do a second PCR off of that PCR. So anything that amplified here, now we can sequence the junction and we can do NGS because this is a short enough amplicon that we can sequence it. Then you can get an output. Basically, if you had an amplicon, you'll get reads, and if you didn't, you'll have a blank well. And so we've just written some micros and some Python programs to basically tell us all the wells that had 5', all the wells that had 3', and then we can figure out which wells have the modification that we want. And in this case, 62% of the clones had both correct junctions with the 5' and the 3' end. All right, so kind of to wrap it up, I wanted to tell you, I told you at the beginning, there's this barrier to entry. I think NGS is super valued. It has a lot of benefits for it, but people are scared to get involved with it. We hope that the expertise required for analyzing the FASTQ files is we've lowered that barrier by CRIS.py and these other NGS analysis programs, but there's still the cost associated with it. A lot of people are like, oh, it's too expensive, I'll just do Sanger. I think you can actually make NGS more cost effective or cheaper than Sanger, and I'll tell you how. The way we do it is the more samples, the better. So we sequence anything we can sequence. Anything that's less than 500 base pairs, we try to sequence with NGS. So we sequence our gRNA plasmid, we sequence pools and clones, we sequence those junctions, deletions, translocations, and we can even do STR profiling to understand the cell line identity. We also pool all of our samples into one run per week. So we get as many samples as we can and we pull them together for the run. You might consider trying a spike-in program if you don't have a lot of samples. And what I mean by this is basically you can get a whole bunch of different samples from different labs. As long as your indexes don't overlap, you can basically pull them together and then parse out the data. This is really economical if you only needed, let's say, so for example, 1 million reads is about $400 in reagents. Whereas if you run a full run, you can get 15 million reads for about $1,200. You get 15 times the reads for 3 times the cost. So the more samples, the better, the more economical. We typically do a 2-step PCR because it's most effective compared to ligating adapters. And you can make your setup quick and easy by doing robotics if possible, or a semi-automatic stampers. We do a crude lysis on our genomic DNA, and we use a multi-drop for fast reagent dispensing. Last 2 slides are in the home stretch here. The cost of NGS can actually be cheaper than Sanger sequencing. And I was telling somebody earlier today, like people in my lab will complain, oh, I have to Sanger sequence something because it's so much harder. It's not really harder, but when you get in the flow of things, NGS really makes sense. So here's an example of how it can be cheaper than Sanger. Like I told you, the MySeq reagent, just the reagents cost, I understand there's a person involved in running the machine and so on, but just the reagents are about $1,200 list price. The number of reads you typically get from a run is about 15 million. If you get 1,000x coverage, which is way more than we actually need for most of our experiments, that comes out to about 8 cents a sample, so super cheap. Let's say you don't need 15 million reads and you don't have a spike-in program. You could run a nano kit. This is about $400. You get about 1 million reads, and that's at 1,000x coverage. That's about 40 cents a sample, so still cheaper than it would be to do Sanger sequencing at most institutions. So be creative, think about it, think about all the things you gain by doing NGS. I don't get paid by anybody who does NGS, but it really has changed our lives. So conclusions, I just wanted to mention that in conclusion, the NGS is an excellent on-target assay for CRISPR activity. It's also, I want to mention that CRIS.py, this NGS pipeline for analyzing NGS, CRISPR edited NGS samples, is freely available. You can get it on GitHub. We also have PrettyCRIS.py out there if you want. And it allows even novice users to interrogate their data. You can really change your test sequences to understand what the editing outcome is. And it's a high throughput method for quickly, cost effectively, and quantitatively measuring many editing outcomes at once. Final slide is I just want to mention the 2 groups that really I presented the data from. The CAGE, which is at St. Jude Children's Research Hospital, and then Genome Engineering, an IPS center at Washington University. I mentioned all the people in red, some of the work that they did. So Monica led the survey of the validation assays, Patrick led the CRIS.py pipeline, and then Sam and Colin helped with the survey experiments. And then thank my funding, and then I'll leave this slide with the people who actually do most of the work, or all of the work, and then these 2 folks, they also joined the lab recently. And with that, I will hand it back over to you.
Thank you, Sandra, for the very informative talk. And while you might be typing in your questions right now, I would like to take the opportunity and introduce a little bit about GenScript's CRISPR services. So actually, we offer sgRNA as well as single-strand DNA, as Sandra mentioned in her talk. So for sgRNA, we offer 2 different grades. We have our synthetic easy edit sgRNA with modifications to support easy and effective editing. It's ready to use, have very high knockout efficiency, even hard to transfect cells, as well as we deliver that at different scales, as well as deliver that in multiple plate format as well. And next slide, we also have safe edit sgRNA, which is high purity sgRNA with modifications, but also HPLC purified. So guaranteeing more than 90% purity to ensure minimum off target as well as cytotoxicity. And this is more designed towards using primary cells or stem cells application. So with a higher purity and less truncated oligos in the sample, you have minimized cell availability and off target. And for lastly, for single-stranded DNA, as Sandra mentioned, we do offer the service, and a lot of our customers are using that actually in cell and animal generation now. And for our single-stranded DNA service is made with no harsh chemicals. It's using enzymatic approach to ensure there's non-detectable level of double-stranded DNA. The length range we offer is from 150 to 5,000 nucleotide in length. We can deliver up to 40 micrograms for flexible study design. And also we have sequence verification done by Sanger sequencing of the final single-strand DNA product. Also a purity test by gel electrophoresis. Also, if you order genes from us, or if you're reordering your past single-strand DNA, we have a lifetime gene template storage to support fast and more cost-effective reordering. And also with our more than 16 or 17 years of experience in synthesizing difficult genes, we can synthesize very more complex single-stranded DNA as well as your HDR template. So yeah, with that, I think we will take more information. You can check about more information at our website at genscript.com, or you can directly contact me for more details at clairez@genscript.com. And now we'll start our Q&A session. Please type in your questions in the question box if you haven't done so already. Thank you.